Indonesia ranks third in tuberculosis burden in the world, with an estimated 824,000 cases and 93,000 deaths per year. Immediate steps are needed to scale up preventative, diagnostic, and treatment services for tuberculosis to break the chain of transmission and reduce the case of multi-drug resistant tuberculosis. Indonesia is also experiencing a rise in non-communicable diseases such as cancer, diabetes, and stroke. And together, these diseases put significant pressure on our health system. Every human has a unique DNA sequence, and those differences can influence our susceptibility to disease and our response to treatment. Understanding these will help us improve prevention, diagnosis, and treatment through precision medicine. Biomedical and Genome Science Initiative BGSi is Indonesia's first national initiative for precision medicine. With priorities towards 5 high-burden diseases and a focus on exploiting genomics to improve our well-being and help us through the aging process. BGSi will support the routine use of genomic information to improve public health using an integrated platform that brings together 3 services units: biobank, registry, and bioinformatics. A biobank is a collection of biological samples such as blood or DNA and health information that is stored in specialized facilities and kept in controlled conditions for future use. Our biobank will collect samples for cancer, stroke, diabetes, TB, and psoriasis patients, as well as from healthy volunteers. A registry is a structured data repository that curates information for individuals that are included in a specific population, including information about the disease, clinical information related to each patient, demographic information, and others where this information can be used for clinical or policy purpose. Our registry will connect this information with the samples stored in our biobank, thus ensuring that these samples can be used for further research and innovation. Imagine being able to use samples collected today for research in 10 years' time. BGSi's Bioinformatics Service Unit will focus on supporting the primary and secondary analysis of the whole genome sequencing data, which would then be used to assess gene variation and expression, and other information generated from next-generation sequencing facilities spread throughout Indonesia. BGSi will work closely with 6 hubs, each targeting different diseases, with the support of 7 national hospitals. BGSi will coordinate the spread of 48 next-generation sequencers to Indonesia's research institutions to increase enthusiasm for biogenomic research. Indonesia has begun its journey towards the establishment of a national genomic sequence reference with a plan to collect 10,000 human genome sequences over the next 2 years, as a collaborative effort to support the complete mapping of disease-specific variants in our population. This project requires us to leapfrog several technological aspects, including next-generation sequencing, bioinformatics pipeline, methods for genotyping single nucleotide polymorphisms, and high-performance computing. Those technologies will discover precise medication for each individual based on their genetic profile to make the best therapeutic choice by facilitating predictions on benefits and/or potential side effects after taking a particular medicine. This can only be realized through inclusive participation and collaboration of various stakeholders. We require your support to ensure we can transform healthcare in Indonesia towards the use of precision diagnostics for more personalized treatment and prevention to improve public health.
In the next few decades, health services will be significantly transformed. Medical treatment is getting more precise by looking at the patient's genetics, lifestyle, and environment. Smartwatch as wearable devices will help us and help workers to monitor health conditions as well as detect emergency situations for immediate response. Patients can access their medical record via mobile phones. This is the time for the technology transformation. Health technology transformation is the sixth pillar of the Ministry of Health's health transformation agenda. Starts with the development of a platform as a service named Satu Sehat, which will integrate all health technology systems in a standardized format using the global buyer standard. Primary, secondary, and private healthcare facility integration. This is the health data interoperability through the Satu Sehat platform. The integrated data will easily be accessed by patients and health facilities based on the patient's consent. Health transformation, Satu Sehat.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Salam Om Swastiastu, Namo Buddhaya, dan salam kebajikan. We welcome everyone tonight to the first BGSi webinar series with the topic NCBI Sequence Variation Database, dbSNP and ClinVar. This discussion is a collaboration between Indonesia Ministry of Health, MOH, and the National Institute of Health, or NIH, US. NIH is a DC-based government body under the Department of Health and Human Service, focusing on the research of health study. And with that, today's discussion will be a very fantastic one. We will learn so much more about the research in NIH. And today's discussion will be conducted in English. And by the end of the discussion, we will have a short Q&A session for everyone. And during the Q&A session, anyone who likes to ask a question can write it on the YouTube chat column. And please note that anyone who likes to ask a question in Bahasa Indonesia can also be facilitated. Jadi mohon izin Bapak Ibu, bila berkenan, kegiatan diskusi akan berlangsung dalam bahasa Inggris, dan bila ada pertanyaan bisa dituliskan di kolom YouTube. And again, for anyone who didn't know, the BGSi is new initiative launched just last year by the Ministry of Health of Indonesia with the focus of sequencing technology and genomics. The project itself is hoping to bring the genomics and sequencing technology to our healthcare and moving toward precision medicine. And today's discussion will be a very exciting one because we have invited two senior members from the NIH, and we will discuss two of the public variation database that one of the largest in the world, and we're looking for engaging discussion that help us to learn more and inspire us to use the database. Our agenda today will be a 20-minute talk for each database and its related research. Each talk will present the purpose of each database, its value, impact, the kind of data it accepts, how to submit it, how it uses, and the API to access the data. And for the data itself, the Digital Transformation Office of the Indonesian Ministry of Health is focusing on such kind of data, from the health data into the sequencing data. And with that, allow me to introduce our first speaker, Dr. Lonpan. Dr. Lonpan has been an NCBI staff scientist for the last 23 years, and he currently the team lead of Human Genetic Variation Group, which oversee the development of dbSNP. The other research he work includes the database of genomic structural variation, dbVar, L frequency aggregator or alpha, variation viewer, variation services, and the dbGap Fire API and the NCBI 1000 genome browser. His research interests include the development of computation and data integration methods to identify novel connection between variants and other data research to enrich the annotation. So with very exciting topic, we will start the discussion in a brief moment. We will start with a short video.
All right. Thank you for the introduction. I'll share my screen. All right. Thank you everybody for participating today. So today I'll be describing you about the NCBI general genetic variation resources, and in particular, so genetic variations, you know, the introduction already described, so I won't go in detail, but essentially it's where you have genetic change between two individuals. So here we're just looking at simple genetic variation, single base changes. You can have single base change or deletion or insertion. But these genetic variations can also span multiple bases, you know, kilobase, megabases in terms of large deletion, insertion, translocation, stuff like that. But today we're trying to focus on this small genetic variation. So do you know the genetic variation are the common variation are, you know, can be effect of phenotypes. For instance, here, this is an example. This is an ad that's using actually 23andMe, which you may be familiar with. They're used for people for essentially for recreational genotyping to look at their ancestry, but they also report certain phenotypic effect or consequence of the genetic variation. For instance, here, this one affect hair coloriness. Of course, the genetic variation can also affect cause diseases, but also they affect treatment and outcomes like drug response, and also they're important for studying human evolution, but they're also used in forensics and so on. So NCBI have primarily two large general genetic variation databases, dbSNP and dbVar. Difference are the dbSNP is for small genetic variation, typically less than 50 base pair, and dbVar for much larger variation, like I mentioned, that could span whole chromosome or kilobase and megabases. So dbSNP is the largest database. It has about over 3 billion submitted records, and those get clustered or aggregated to unique variants. And we assign them what they call reference SNP numbers. And so there's, and these are used for annotation and for data sharing and propagation across resources. So this, if you look at this, essentially, you know, there's one, on average, there's one genetic variation every 3 to 4 base pair in the human genome. So, and then the other, I guess, sister database or project to dbSNP is the Alpha project. So this is a newer project that we initiated a few years ago. So this takes advantage of all the genomic data that's in dbGap, and so we aggregate that and provide a low frequency for the variant in dbSNP. Okay. So I'll briefly go over the session and the submission process and the how we generate the reference SNP RS numbers. So typically the submissions are coming more recently, they're coming as VCF. So it's a very simple process. We typically take the VCF that come up directly off the your variant calling pipeline. So there's very little modifications for you. So it's very simple. So just in the act, just VCF to us. And we accept submission from all around the world. It could become from multiple projects, from multiple populations and samples, something like that. So like I said, we have over 3 billion of these, what we call, so each variant that comes in, we assign an SS number or a submitted SNP session. And then those go to our pipeline, and then they get clustered. So essentially, variants that are identical will get the same a cluster, the same RS numbers, and they're clustered based on variant type and genome location. So, and then in addition, we also take submission for allele frequency data. So it could be from different population projects. For instance, here we got the Alpha project that's included that I already mentioned, down genome, TopMed, NOMED, and hundreds of other projects that we have in dbSNP. There may be specific population like Vietnamese, Finnish, and so on populations. Anyway, so we assign RS numbers, and so 3 billion get reduced down to about 1 billion. So then the RS numbers are their unique session that can be used for exchange and citing and stuff that. They also used to annotate on the human genome and RefSeq sequence, the mRNA and protein. And then the data then make available to the public. There's a web search. There's often, we have an API. Actually, there's 2 APIs. There's one that the EUTIL, which you may be familiar with, that's general. So you can access the data, dbSNP data, to the EUTIL API or our variation service API, which I'll describe a little bit later. And then, but typically because of a large amount of data, most Biomedical pipeline will download the data as VCF or JSON format from our FTP site. And also the data is also, you can also view it on the genome viewer. And then user can download data, you can integrate into their normal Biomedical pipeline. In addition, so our various service, we have, you can retrieve our RS number, but it's also have a couple of utilities that may be important to you in terms of it can help normalize variants and convert it between different notations so that you can identify the same variant that are described different ways. Like I mentioned, we typically take submissions in VCF format that they have the sequence, position, the reference base, and the chain base chain. And then we aggregate that, generate RS numbers, but then that get annotated in different sequence, and then we generate HVS expression for each of the sequence. And we report the variant on all supported GRC assemblies. For instance, here is showing to you for both the older versions, GRC 37, and also GRC 38, and the different NM or mRNA RefSeq versions. And also, so you may be familiar with this, and then what's new is the NCBI speedy format. So we developed this mainly because it's facilitate computation. Because it reports the variant, it's somewhat similar to HES. You can see there's some similarity, but it's report on absolute position rather than relative. Like for instance, here, this is relative to coding sequence start or something like that, and then whereas we support the absolute. So when you do computation, there's not a whole lot of conversion. Is very fast. You just use directly. You know what this at this position, just change on this sequence. You also support some other external IDs conversion, also like the GFA, the variation representation model, GH for ID. But anyway, so that's why I went. But if you want to read more, there's a blog, there's a link on here about the describing speedy and the variation services. Okay. So once the RS is generated and annotated on the sequence, and then it's released on the public. So this just came out recently, Bill 156. So we report, this position, the allele, the variant type, the frequency, and if there's any clinical significant reported in ClinVar, you can click on the link there, which pick the ClinVar that Melissa will tell you about in a little bit. We always report you the gene it's mapped to and the consequence. For instance, it's a missing variant, any publication associated with it, and see it on the sequence viewer. There's also additional details here, such as frequency, variant, and other details about the variant that you can investigate. But first, before we go into the frequency, I can describe you how we generate the alpha frequency data and about that project that goes into dbSNP. So alpha stands for low frequency aggregator. So this is a new source of frequency data or genomic summary result from dbGaP. If you're not familiar with dbGaP, so it's one of the world's largest database for storing studies that look at the association between genotype and phenotype. So it had a lot of, genotype, GWAS data, array data, and stuff like that. So there's over 2,000 studies, and there's over 2 million subjects. So it's a huge pool of data that we can use. So this allows us to aggregate the studies and then generate the allele frequency for dbSNP. So here's the workflow essentially for, or the data flow for alpha. So we start with selected dbGaP study that have been approved for summary results or open access. And those, this is from our last release 2. So this is just a small number of studies, right? So it's about 80 studies, about 200,000 subjects, and about 6 trillion genotype. So that goes into a pipeline, like, again, kind of like similar to dbSNP. They get clustered, all the variant same location, and they get assigned RS numbers, and either to existing RS number or get assigned RS numbers. And then we then aggregate across all the samples and subjects and compute the allele frequency, and then provide output as RS numbers and unique variants. And so the 5 trillion genotype get aggregate down to almost 1 billion RS numbers. And then again, we provide the data as open access. There's just like dbSNP, there's FTP download, and then also it gets integrated into dbSNP resources. So if you're already using dbSNP, the alpha data essentially come for free because it just follows the same. It's embedded within the dbSNP data object. So we generate low frequency for cell 12 population, typically. So these are large continental population, European, African, Asian, and so on, Latin American. So there's about 200,000 subjects. Most of it is European. So we would like to get more of the underrepresented population, like for Asian for instance. As I mentioned, alpha is the world's largest open access research source compared to other projects. For instance, TopMed, you can know, and NoMed. So even with just less than 10% of dbGaP data, and only on our second release so far, we already exceed TopMed and other projects in terms of number of subjects, but also number of variants. So we are close to 1 billion variants. Right. We're anticipating the next release will come out this summer, and that will probably be close to 250,000 or 300,000 subjects. Okay. So the typical usage or use of our data is in both for research, but also in a clinical pipeline in terms of for detecting variants are related to or associated with diseases. So typically for variant, the data we use for variant discovery project, for let's say if you sequence, you sequence a bunch of subjects, you call out a variant, you see, well, which one is novel. So you can use dbSNP to filter, which one is novel and not that exists in dbSNP. Or you can look at, only uninterested and common variants. So with the frequency data in dbSNP, you can use that to filter for common and rare variants. And in addition, there's other, there's probably another dozen more, but I just find 2 examples here. And you can look at, filter further down to missense or non-synonymous variant, and those that are pathogenic. So so for just this simple filter, you went from over 1 billion variants down to a few thousands. course, then you can filter by genes and then so on like that. And then that will get you down to a handful of variants. Okay. So dbSNP is been in existence for, this will be dbSNP 25th year anniversary. So we will have some celebration and activity around that. But it's been used for a long time. So it's generally recommended as best practice to use in a lot of different resources in terms of for genome pipeline, it's also incorporated into Ensemble or EBI tools, there are API, there's open source tools. It's also used in variant column pipeline. Again, it's to validate or to identify novel variants. Okay, and it's also a guideline to filter out common variants for if you're looking at pathogenic variants. It's also popular, like I said, it's popular in various different resources and been used. The RSS session is cited common almost everywhere. And it's, for some, you may be familiar with some of these resources. There's SNPpedia, UCC Genomic Browser, and some open source project down Genome and such on. So the RS number has been cited over 60,000 publications. For instance, here is an example where these variants have been identified, or RS number identified as affecting, perhaps involved in cancer. And it's actually looking at in Malays populations. So if we look at this first RS number in dbSNP, you can see here, for instance, this is the alpha frequency reported for it. And here in European, it's reported as common, and whereas in Asian is rare. So that may affect how you interpret that variant and for your population, right? So with that, that's kind of a brief, quick introduction of the research at NCBI, and I'll be happy to answer any question.
Yeah,
Okay, thank you so much, Dr. Lonpan. Obviously, the dbSNP is the first go-to database for someone who worked in genomics and also bioinformatics. And if I can summarize, the dbSNP is a public archive for genetic variation across different kinds of species hosted by the NCBI. The database itself can be accessed in many ways, such as web access, FTP, and API. We can also access different kind of data formats, such as VCF, JSON, and other. The dbSNP is considered a best practice available today as a database to annotate genetic variations. And with that, I think I would like to a little bit discuss about the dbSNP. For someone who worked with the bioinformatics, everybody should know the dbSNP along with its uses. But for the last 25 years, the dbSNP has been used and publicly available online. Obviously, the dbSNP grows every year, every week, for including more and novel data to add to its database. But with the advent of third generation sequencing and also single cell sequencing, how much the growth of dbSNP we expecting for the couple of future of years?
Sorry, your question that you expect how much growth in the future?
Yeah, because I think the next generation sequencing will give us a better, enable us to map our genome a little bit better, as we can understand more previously difficult to sequence region.
Right. So dbSNP, I forgot to mention, is that so it's a general generator to store both common and rare variants. And also there may be some mutation in there from, various variant complex, because again, people are doing, whole genome sequencing of individuals. So right now, we don't expect the whole, I think probably most of the variants have been discovered. So there may be a slight bump here and there as we sequence more subjects, more like, so I would say most of the common variants have been found, probably around 85 to 90 million. Actually, most of dbSNP is now almost 90% of it is rare variants. And most of these are sort of like, the private variants or the personal variants that people that have been discovered through various whole genome or genotyping projects. So in terms of numbers of RS numbers, I think we're, because of the data, most of the data we include from alpha is from the last release is from whole genome sequencing. So I think most of those variants are already been found. And so there may, like I said, there may be a small bump in terms of the rare, super rare variants, in terms of private variants or specific cohorts and stuff like that. In terms of content, we're still constantly looking for in terms of annotation. I think that's where the growth would be in terms of, we may be looking at including, let's say, functional elements like enhancers or promoters and integrating other resources like annotation for function and stuff like that, and clinical information and publication information.
Thank you so much. And for the dbSNP ecosystem, so to say, there's a number of research related with dbSNPs. There's dbVar, alpha, and so much more. How this kind of research interact with each other? Is it with the Fire API that your team has been working on? And it's quite relevant with the DGO itself, especially Indonesia Ministry of Health currently are working together to establish the standardized health data, including the use of Fire. How the implementation of the Fire and dbSNP interact with each other in the NIH?
Right now, so Fire is a fairly new project that we just started just for dbGaP data. We haven't implemented, the issue with Fire is that there's no representation for variants yet. And it's very difficult to exchange genotype and that kind of molecular data right now in Fire, right? So it's more oriented more like health records. So if that, in the future, if that representation is expanded, we could possibly, yes, include a service or something like that that represents dbSNP and Fire for exchange. But right now, there's no plan to do that in the near future. In terms of because also in terms of the BOM data, you're talking about over 1 billion variants. Typically, our data flow and it's around NCBI is just moving files and database around and not using APIs.
Okay, fantastic. And for anyone who currently watching, the Indonesian Ministry of Health currently are working together to push our project specifically in genomic and sequencing technology with the BGSi, Biomedical Genome Science Initiative. And we're hoping that we can learn so much more how the health data and the sequencing data could help each other and working toward data-driven healthcare. Okay.
Yeah, we'll be happy to help you integrate the data within your resources.
Okay, thank you so much. Thank you so much, Dr. Ranpan. And with that, we're moving on to our next speaker, and I will read her short bio. So Dr. Melissa Landrum is the team lead for the ClinVar database. ClinVar database itself is a product of National Center of Biotechnology Information within the National Library of Medicine of the National Institute of Health. Dr. Melissa has been working with the NCBI for the last 22 years, leading the ClinVar team for the last 10 years. And prior to that, she was a curator for the NCBI reference sequence database, RefSec. Before joining the NCBI, Dr. Melissa earned her PhD in human genetics from the Johns Hopkins University. And with that, time and place, it's yours.
Great, thank you all for having us here today. Great, thank you all for having us here today. And I am happy to tell you a little bit about ClinVar. Do you see my slides?
Yes, we can see it clearly.
All right, great. So I'm going to tell you a bit today about ClinVar, which is a worldwide archive of variant disease relationships. So as a bit of background, let's talk a little bit about why genetic variants matter. So you can think about them being important in two major realms. One, of course, is clinical genetics, where variants may have no effect at all, or they may have an innocuous effect, so they affect your eye color or your height, but not something that really affects health or disease. Then there are variants that have a deleterious effect and they cause a disease, or there are variants that affect how a person responds to a drug. The second realm where variants matter is within research. So of course, variants are used for studies of population genetics, evolution, and other kinds of basic and clinical research. So dbSNP, the database that you just heard about from Lon, holds variants that would be useful in all of these ways. But in ClinVar, we're really focused on the variants that are important for clinical genetics, so those that affect disease or drug response. There are also variants from research, but really only as they inform clinical genetics. So anything in research that would help those in clinical genetics assess a variant. So as we are focused on clinical genetics, let's talk a little bit about the big challenge for this field. So the sequencing technology that we have today makes it very easy and fast to identify variants. But the process of determining which variants are innocuous versus which variants are important for disease or drug response takes much more time and effort. So there's a process called classifying variants or interpreting a variant to decide if the variant's important for health. And that requires a good deal of research and collection of evidence. And the collection of evidence can be a big challenge because many variants are rare. Any individual laboratory may only see a rare variant once, so it can be hard for them to gather enough data to make a good classification of that variant. So that's why sharing data among laboratories is critical so that all labs can improve their ability to classify variants accurately. And those accurate classifications of variants are critical for patient care so that physicians and patients can make a good decision based on the patient's genetics. So ClinVar aims to help this challenge by aiding data sharing. So ClinVar is an NIH database of classifications of genetic variants and how they relate to diseases and drug responses. So the data that you see in ClinVar are considered variant level classifications. We'll talk a little bit more about that in a later slide, as opposed to patient level classifications. And the database is driven by submissions. So laboratories and other groups voluntarily share their data with the world through ClinVar. And then we make that data public and available for anyone to use when they go to classify variants. And it has truly become a global database. Today, we have more than 3.25 million classifications for more than 2 million variants. We always have more classifications than variants because some variants are classified by more than one laboratory. And these data come to us from more than 2,400 submitters from 87 countries across the world, as you can see. Indonesia is not on our map yet of countries that participate in ClinVar, but I can tell you, I believe we have our first submission from Indonesia. I believe it's a research laboratory, and the submission is being held until the corresponding journal article is published. So we're happy to welcome Indonesia to the map very soon, and we'd really love to work with you all to get more representation from your country too. So before we go into too much more detail about ClinVar, I do want to talk a little bit about ClinVar and ClinGen, a group that we work closely with, both to help you distinguish between the two groups and help you understand how we work together. So NCBI's ClinVar is the database we're talking about today. So this is the archival, submission-driven database that's created and maintained by NCBI, and we are supported by intramural funding at the NIH. ClinGen is the clinical genome resource. So this is a database of expert curations. It is created and maintained by ClinGen, which is a consortium with members from multiple institutions who are funded primarily by grants from the National Human Genome Research Institute, the NHGRI, also with funding from a few other NIH institutes as well. And one of their main activities is expert curation in these 4 areas of variant disease classifications, gene disease validity, gene dosage sensitivity, and clinical actionability. And it's the area of variant disease classifications where we have a very close partnership with ClinGen. They're developing expert panels who do this expert level curation of variants, and those expert curations are shared into with ClinVar. So as I mentioned, ClinVar is this archival database where our function is to collect data and then provide it back for use. And we collect data from many sources. So most of the data in ClinVar comes from clinical laboratories who share the variants they classify as part of their work doing tests. We also have data from research labs. Data comes to us from expert panels. So that's the partnership we have with ClinGen. They're developing most of the expert panels. We also get data from patient registries, which tend to have more phenotype information than the clinical labs have. And we also get data from curated sources like gene reviews and OMIM. So these groups submit their data to ClinVar. On our team accepts the submissions, we validate and standardize the data, we aggregate the information, and then make it available on our website through FTP and through APIs. And then the data is used by various groups, including clinical genetics testing labs, clinical genetics specialists like medical geneticists and genetic counselors, other clinicians who might order genetic tests, researchers, and other groups. So because we take in data and our data is used and by many different kinds of groups, it's important that we use data standards to make sure that the communication is clear. So we use standards whenever we can. So for variants, we use HGVS nomenclature. We support identifiers from dbSNP, dbVar, and the ClinGen allele registry. We also assign our own identifier called the variation ID, which has now become a standard for reporting in lab reports and in publications. For genes, we use the official gene symbols that are assigned by the Human Gene Nomenclature Committee and the identifiers that are assigned by the NCBI gene database. Disease and phenotype is a very complex field, so we support several data standards here. 4 of them are shown on this slide. We try to map all variants in ClinVar to reference sequences. So to map variants to the genome, we use the genome assemblies that are maintained by the Genome Reference Consortium. We support both the previous assembly, GRCH37, and also the current assembly, GRCH38. Most of the variants in ClinVar are within a gene, so most variants can also be mapped to a transcript, the appropriate transcripts for that gene. So we use RefSeq transcripts for that. We also indicate which of those transcripts are the main transcripts. In other words, which transcripts are recommended for clinical reporting. Within ClinVar, we annotate variants with information like where is the variant within the gene? Is it predicted to be missense or frame shift? So there are a couple of ontologies listed here that we use to provide standards for that kind of information. And then finally, the classifications themselves have standards that are being developed. You may be familiar with the ACMG AMP guidelines that came out in 2015. These are guidelines for interpretation of sequence variants, germline sequence variants for monogenic disease. So we use those standards. There's also a standard that came out in 2017 from AMP, CAP, and ASCO for the classification of somatic variants, and we are working on incorporating that standard into ClinVar now. So I mentioned earlier that there are lots of kinds of groups that use ClinVar data, but the two groups that we are most focused on helping are clinical genetics testing laboratories. So those groups who are classifying variants and generating lab reports, and also clinicians. So specifically the clinicians who are working in genetics, so those who receive the lab reports and must make sense of them for their patients. So it's these two groups we're most focused on helping. And you can imagine a couple of use cases. One is from the perspective of the clinical testing laboratory. So let's say you are that laboratory and your lab has identified a new variant. Some questions that you might come to ClinVar to answer are, have other laboratories seen this variant before? If so, did they classify it as disease causing, and for what disease? And importantly, what was their evidence for the classification? This is where it becomes useful for different groups to pool their evidence, and they may come up with a better classification. Another use case is from the perspective of the clinician. So let's say your patient has a genetic testing result. Maybe the report says that there's a pathogenic variant or maybe a variant of uncertain significance of the US. Questions that you as the clinician might come to ClinVar to answer are, how have other labs reported this variant? Did they agree with the result that's on my lab report? What phenotypes were present in other individuals with this variant? Again, is it consistent with what I see in my patient? And then finally, is there new evidence for this variant since my patient was tested? For the clinician, they will see their patient repeatedly. Maybe they come in for yearly visits, and each time they might come to ClinVar to see, is there new information since the last time I saw my patient? Do we need to change clinical care based on current knowledge? So you can imagine any of these groups, maybe a testing lab is generating a lab report with a variant, say in the gene G6PD, the variant is P459S, or maybe you're the clinician who has received a lab report and you see this variant. So any of these groups can come to ClinVar, put in this kind of information. Whatever information you have in the lab report can be entered into ClinVar. We support several types of searches. And you can find information about that variant. So this is a typical variant page in ClinVar. The top of the page shows you the variant that you're looking at and some summary information. So here you can see this variant is considered pathogenic based on 2 submissions to the database. There's some more details about the variant itself. And then as you scroll down the page, you can see the details of each submission to the database. So in this table, each row is a submission. So you can see we have one report that the variant is pathogenic. This came to us from ARUP Laboratories. This is a clinical testing lab in the US. And we have another report that the variant is pathogenic that came from a research laboratory at the University of Washington who provided some curation results from their work. So there's a few things to notice on this page. One is that you'll see that individual variants are submitted to ClinVar. It's not whole genome data. So in this case, each laboratory submitted their classification of just this single variant. So we don't have the same issues of privacy and identifiability that are associated with whole genome data. Secondly, I mentioned earlier, the classifications that you see in ClinVar are variant level classifications, not patient specific. So in each case, you can see from the comment from this first submission and from this information about the condition in the second submission, the variant is classified for a disease. So each of these groups is looking at the relationship of this variant to anemia associated with G6PD deficiency. It's not specifically describing if this variant explains a specific patient's phenotype. It's what do we think this variant means for a disease in general. The evidence that's submitted to ClinVar is typically aggregate and de-identified. If you read through these comments, you can see they are generally describing their data, but not giving specific information about patients. That's the typical case. Sometimes details about patients may be submitted But this is only if the submitter has consent to share that level of information, similar to publishing patient information in a journal. And even when the submitter does have consent to provide that level of information, we still ask that no personal health information be included, because we do make the information fully public to anyone who wants to use it. So to prepare for this talk today, I did go look at the BGSi website to see what sorts of things you're interested in and how ClinVar can help you. So I looked at your page on genetic disorders and noticed that 2 diseases you're focusing on initially are Duchenne muscular dystrophy and pulmonary arterial hypertension, or PAH. And related to PAH, I saw that you mentioned one gene, BMPR2, has pretty good support, and there's several other genes that have been implicated, but maybe need more investigation. So this is a place where ClinVar can help you in your work. So for example, let's take the gene SMAD1. You might come to ClinVar to investigate what's known about SMAD1 and PAH. I did this search last week. Here I'm looking for just SMAD1 as a gene, and I narrowed down the results to just variants that are within the gene, so no large copy number variants that span many genes. And you can see in the diagram here, there's a few dots. It represents a few more variants than you see here. Some of them are clustered, but it's a handful of variants. And you'll notice that all of the dots are green. So that means that these variants have been reported as likely benign or benign. So far, we don't have any variants that are thought in SMAD1 that are thought to cause pulmonary arterial hypertension. So this could inform your work in a couple of ways. You might look at these results and say, there's not very good evidence for involvement of SMAD1 with pulmonary arterial hypertension. Maybe we should focus our work on some of these other genes that have been implicated. Or alternatively, if you have really good evidence in the Indonesian patients that maybe SMAD1 is disease-causing for PAH, you can tell from this that that would be very valuable information to get into ClinVar, because others have not reported that yet. That would be new information that would be valuable to the clinical genetics field. So the data in ClinVar is accessible for you to use in several ways. I just showed you can search for data on our website. You can download bulk data on our FTP site. The XML files are the comprehensive data set, but we also have summary data available in VCF format, as well as some tab delimited files. And you can also access the data programmatically by API. We use utilities, which Lon mentioned earlier. This is a standard set of API functions that are used throughout NCBI's Entree databases. So in addition to using ClinVar data, we'd also be happy to have you all submit data as well. We think we can, as you can see from the map I showed you earlier, ClinVar is a global database. We are starting to get data from more diverse sources than just the US clinical testing laboratories. And we think having data from many sources across the world is really valuable in a couple of ways. So diverse sources of data can contradict or sometimes correct existing variant classifications. So the example we're looking at here is a variant in the LDLR-AP1 gene. We had classifications from a couple of laboratories which had thought that the variant was benign, but we had another classification from Illumina who had classified the variant as uncertain significance, which meant they thought there was a chance this variant could cause disease. So in 2021, we got a submission from the H3 Africa Consortium, which introduced new data to the pool. And as part of their work, they looked at the frequency of variants within African populations. And so for this variant, they found that the frequency was greater than 5%. If you're familiar with the ACMG guidelines, if the variant is more frequent than the prevalence of the disease, it is highly unlikely that variant can be causative for the disease. So this kind of information is really strong support for the variant being benign, and it can help a lab like Illumina maybe reconsider their classification and reclassify this variant. Another way that diverse data sources can be valuable is by contributing novel genomic information. So here we're looking at a variant in the COL-Q gene. You can see it's thought to be pathogenic and just based on a single submission. So in this case, say a medical center in Malaysia submitted this variant that they think is pathogenic for congenital myasthenic syndrome 5. And it's based on a single person that they did clinical testing on. So you can see, I mentioned earlier, many of these variants are rare, and the only time we're going to see it is if that one laboratory that tests that one patient shares the data with ClinVar. So it can't be understated that every submission that comes to ClinVar is valuable. So we support submission in a few different ways. We have an online web form or a wizard that walks you through submitting one record at a time. You can see there's a set of tabs here that walk you through the different types of data. This is a very manual process, so it's good if you have a very small number of variants to submit or if you're an infrequent submitter. We also have bulk submission in files. So these files are either our Excel submission spreadsheet templates, part of which is shown here, or you can submit tab or comma separated files that are based on this template. This requires a bit of investment to map your data to our template. So this is useful when you have more than just a handful of variants to submit, or you're going to be a repeat submitter. Most of our data comes in on these Excel spreadsheets. And then finally, we also have an API for submission. This is good for those who have large volumes of variants to submit, those who will be repeat submitters and who have programmatic support. Currently, these are typical clinical testing laboratories who submit regularly to ClinVar large volumes of data, and they have a bioinformatics team who can support them on their end. So finally, I just want to thank the team that supports ClinVar. We have a small team, but we do a lot with it. This is our team of developers, our curators, and our leadership within NCBI and the NLM. And with that, I'm happy to take questions as well.
Okay, thank you so much, Dr. Melissa Landrum. So obviously the ClinVar has been a go-to database for someone who works in genetics, especially in clinical genetics, genetic counselor, molecular biologists, and many more. And if I can resume our discussion that ClinVar is a database that focusing on the classification of disease and its relationship. The ClinVar itself is a submission-driven database, which anyone who can have the data can upload their findings. The database itself have 3.25 million classifications. The data can be accessed with different kind of format, such as HGVS and IDs from the dbSNP. ClinVar help clinician identifies variants and its relationships. And with that, I think I would like to start a discussion a little bit about ClinVar. Obviously, the Indonesian Ministry of Health is trying to improve our work in genomics, especially in clinical settings. So currently we have eight different hubs specialized in 7 different kind of disease, which hopefully can improve our understanding in Indonesian genetics in relation to disease. And with that, we hopefully increase our submission from Indonesia, especially to ClinVar. But with that, what kind of validation are required to submit to ClinVar? Let's say we found a unique variation that strongly relates to some kind of phenotypes from targeted sequencing. What the validation we need to do before we submit those findings? Is it we need to go further, like whole exome or Sanger sequencing or other kind of methods? Yeah.
That is a great question. So ClinVar does not enforce any kind of validation in that sense. We leave it up to our submitters to decide how much validation they would like to do on their end. We do validation of the data itself, so we will confirm that the variant description is something that we can identify, that there are allowed values for what you're describing. But we do not confirm the classification itself. The sharing of data in ClinVar, making it available to the world, letting other people see your information and pooling information is what will get us to consensus on the classification itself. If how much confirmation you'd like to do with the variant itself, if you want to check with an sorry, another sequencing method, but like Sanger, I think that's typically each laboratory kind of decides what their validation process is for that.
All right, thank you. And again, with taking focus on Indonesia, we have a very incredible diversity, both in genetics and many other. In relation with genetic diversity from different kind of populations, is ClinVar equipped with such kind of metadata to enrich the data stored in the ClinVar, such as say Genomat or any other database in relation with the population of data submitted to the ClinVar?
So we do have a field where you can indicate the population group or ethnicity of individuals with the variant. If you have that information and you have consent to submit it, we very much welcome that information, particularly because in this country, our clinical testing laboratories typically don't have that much information about the patient. So if your laboratories do have that, that would be extremely valuable to have. We typically pull in Nomad data into our web displays. So we can show what the frequency is from Nomad or Alpha, the population resource that Lon described today. But we do not necessarily ask for that in submissions. We pull it in automatically. Some of our submitters will reference population frequencies and some of these other databases as part of the evidence for their classification, but it's not required.
Okay. Okay. Thank you. Thank you, Dr. Melissa. I think we will moving on to the Q&A session. And for anyone who would like to ask a question, could type his or her question in the YouTube comment session. Jadi Bapak Ibu yang ingin bertanya, dipersilakan untuk mengajukan pertanyaannya di kolom pertanyaan di YouTube. Okay, I think we have a question from Pak Ahmad. The question is: the interpretation of genetic variation is the interpretation of genetic variant annotated in ClinVar consistent with ACMG guideline, or should we be more cautious in taking ClinVar interpretation in making variant interpretation?
That's a great question. So it can vary from submitter to submitter. And so one of the things that we ask for in submission is what we call assertion criteria. So this is a document that describes what classification rule the submitter used to make that classification. So many submitters do use the ACMG guidelines, and you'll see that listed for many submissions in ClinVar. Some laboratories have their own set of rules. It's often they've customized the ACMG guidelines for the specific gene or disease that they're testing, which is expected. That was the intent of the guidelines. Other groups don't describe their rules at all. So it can vary a good bit. And so I do encourage you, when you see data in ClinVar, to look and see what is listed as the rules that they use for classifications. And that can help you understand, how much you want to trust that classification.
Okay, fantastic. Moving on to the next question from Muhammad Rifki. How exactly do the expert curation works in ClinGen? How do they compare to ClinVar curation, and what does this say about the way we should interpret both database?
Another great question. So one clarification I should make is that within ClinVar, we don't really do any curation of the variant classification. So we accept submissions, we aggregate them, we do some calculations to let you know if everybody agrees or disagrees on how a variant is classified, but we are not actively classifying variants ourselves. So In contrast, ClinGen is establishing expert panels, and they take in data wherever they can get it, from ClinVar, from publications. They share private data. You know, that laboratories may have patient level data that they can't share publicly, but they can share it in small groups as evidence for classification. And so that's how they get to do that. And they're also, their goal is for each expert panel to get the world's experts in that gene or disease area. So they're really focused on getting the best quality, most well-supported classification for variants as they can, and then sharing that in ClinVar. So it's a very complementary set of activities that we do. There's no conflict in their curation versus our aggregation of data, if that makes sense.
Okay, thank you. We have another question. I think it's specifically for dbSNP. How big is the data accessed through the web browser and API, especially for the dbSNP?
I don't have a specific number off the top of my head, but I think we get, probably somewhere close to 1 million requests, 500,000 requests per month. Like I said, most of the, because a large amount of data, there's two usage. One is if you're doing a pipeline or something like that, it's best just to download from the FTP site, the bulk download. And then there's the medium-sized user, typically from a couple thousand to a few 100,000. Those use the API, and then the web request, and then the web browser, yes.
Okay, thank you so much. Baik, mungkin kembali mengingatkan Bapak Ibu yang ingin bertanya bisa menuliskan pertanyaannya di kolom komentar. So for anyone who would like to ask a question, even in Bahasa Indonesia, can type his or her question in the column, question column. I think we have a question. Anyone who can answer this, I believe. How accurate the result of Indonesian genomic analysis carry out using currently available reference genome? Hopefully, it is quite relevant.
I could try to answer that. So right now, I mean, in particular, because in terms of dbSNP, so we support all assemblies and all alternate assemblies. So as the sequencing improve and assembly improve, of course, our annotation will improve, and we'll have coverage in terms of the missing region, like the centromere, telomeres, and previously hard to sequence, especially in repeated regions. But that number of variants in those regions will increase, and then also the annotation quality will improve also as sequencing technology improve, the assembly improve, and so on. And in addition, the other thing I want to mention is that the variant itself will be more and more will be validated, because you observe in population and we back again with the growth of dbSNP. I think the growth of dbSNP not is so much the number of variants, but the annotation and data attached, like the frequency data. What we want to do is want to collect more frequency data from more populations, so that's the world, get on board with the personal medicines and sequence more. Hopefully we'll collect some of that data, including from Indonesia, population-specific frequency data that include in dbSNP. And of course, we're also mining that data from alpha. So I think and then the other thing would be as more technology improve in terms of functional assay for these variants, where they're doing genome-wide mass array, biochemical assays on these things. We'll hopefully add that to improve the interpretations and the quality of the variant.
Okay, thank you so much, Dr. Lempan, for the answer. I think we will close the Q&A session. If anyone Okay, so I think we will close the Q&A session and try to summarize today's discussion. So we have explored the many aspects of dbSNP as a database, as a go-to database for human genetic variation, and also we learned so much about ClinVar as a resource to help clinicians classify genetic variation in relation with disease or conditions. And it's clear that from our discussion today that this initiative have significant implication, hopefully to push our effort in genomics and sequencing technology in Indonesia, because we just started with our latest initiative, the BGSi, that hoping to bring more genomics into our healthcare. And with that, I think we will close the discussion for today. I would like to extend my thank to our speakers today, Dr. Lonvan and Dr. Melia Salendram, and the NIH as general. Once again, I would like also thank all the participants for their valuable contribution to this discussion. I look forward to continuing our work together with the focus on genomics. I think that will conclude our discussion today, and hopefully we will have more discussion in future webinar series. Thank you so much.
Thank you.
Thank you.
Bye.
Thank you. Thank you so much. Thank you, Dr. Lonvan. Thank you, Dr. Melsa. Thank you, Karen. Thank you, the NIH team.
Yes, thanks for having us. This was great.
Tuberculosis burden in the world, with an estimated 824,000 cases and 93,000 deaths per year. Immediate steps are needed to scale up preventative, diagnostic, and treatment services for tuberculosis to break the chain of transmission and reduce the case of multi-drug resistant tuberculosis. Indonesia is also experiencing a rise in non-communicable diseases such as cancer, diabetes, and stroke. And together, these diseases put significant pressure on our health system. Every human has a unique DNA sequence, and those differences can influence our